I'm Francesca Puppo from the Mutri Lab, and today I'm going to talk about organoid intelligence. So this new emerging field that tries to use brain organoids for computation. Sorry. Okay, in the Mutri Lab, we model the developing brain using human stem cells. And in the past 10 years, we have been using these 3D structures called the brain organoids to study the mechanisms of the brains. So brain organoids are very big clusters of cells, stem cells, that differentiate into the major cell types of the brain. And they mimic very well the development of functions such as plasticity, activity of the brain. I just want to highlight a few features of these objects. So the cellular composition of brain organoids is very heterogeneous. They contain the major cell types of the brain, and this correlates very well with the activity and with the emergence of oscillations in brain organoids. So we have, amongst the major cell types, we have progenitor cells and glutamatergic neurons at the beginning that give rise to very slow activity and very random activity. Then when, at the age of 3 months, more glutamatergic neurons, the glutamatergic neurons increases, and so we have increasing in frequency. By the age of 6 months, we have the appearance of astrocytes and glial cells that boost the activity further, and we start seeing synchronization. And then between 8 months and 10 months, we can see also a population of GABAergic neurons, so inhibition, which we know is very important for the coordination of activity and of the network. So here, at the end, we see oscillations. So in the previous work, we were able to show oscillations with many, like nested oscillation with very complex frequency that was comparable to the frequency bands of EEGs of preterm babies. But now what we want to do is trying to communicate to these organoids and how we can do that. So in our lab, we stimulate the organoids, and we can do that electrically or with optogenetics. Optogenetics has the advantage of targeting specific cell populations, so we can play on the excitation and the inhibition balance. But we had to improve our transfection protocol so that we have a way to deliver optical probes, optogenetic probes to the organoids. Here we see a very nice confocal image. It's not clear, but that shows the internal part of the organoids with expression everywhere of channel rhodopsin. When we have, when we have these expressing organoids, we can play them on MEAs, so multi-electrode arrays, and thank you. And we can, thanks to the Lumos system that we have in the lab, we can have in parallel stimulation of the organoids with blue lights or any wavelengths that targets the optogenetic probe, and recordings. So here on the right side, we see an example of an experiment where I was simulating the organoids with different frequencies, different intensities, different duration of the pulses, and I could see a different behavior of the organoid. So with this, we have a tool to study plasticity mechanisms, because the standard way to study plasticity is to use high-frequency stimulation. And among all the different paradigms that we use, this was the best that we could use. So a sequence of 30 Hertz trains of pulses with 10 seconds interval in between, repeated for how long we wanted. So in the first graph, we see just a 30 seconds high-frequency stimulation, and we see that already after the stimulation, right after and 1 hour after, the spiking rate of the organoids is increased for some of the wells, decreased for some others, and it's kept higher than the baseline also for quite some time. So this suggests that we have some events, some plastic effect. But then we stimulated with 1 hour high-frequency stimulation. And at that point, we could really see a retainance of the spike rate for 1 hour, 2 hours, or longer after the stimulation. We also looked at the population spiking and the network events. And it was very interesting to see that the shape of the network events was changing if they were detected before or after the stimulation. Before, these network events were showing a very low amplitude and they were wider. And then after, they were very high in amplitude and narrower. And this is very nice because it means that something is happening at the network level, that there is more synchronization, more coordination, and a lower threshold of excitability of the entire network as an effect of the stimulation. So now we want to use all these tools that I just described to implement the organoid intelligence paradigm. So we have the organoid, we have several inputs, and like electrical stimulation, optogenetics. We could even connect retinal organoids to have more complex inputs. And we have the outputs, like imaging or electrophysiology recordings. We are missing the biofeedback loop and the computation, like the AI or machine learning computation that can process, decode and code the signal throughout the whole process and train the organoid. Because in the past, we already showed that our lab, in our lab, we showed that we could connect the brain organoids to a non-biological system. So we showed that the activity of the organoids could control, can control a robot in terms of speed and directions. But this is not an intelligent task. So we don't have any task. We don't have organoid intelligence. So what we are trying to do now is to close the loop. So we would like to give a task to the robot or any other like non-biological system. And then we want to see if the task is accomplished, we want to send a reward. If it's not accomplished, we want to send a negative feedback. But what we are struggling with is how to encode the reward, how to communicate with the organoids. We don't know what is a positive signal for the organoid, what is a negative one. So what I've been working on is to study really the learning mechanisms of these objects, these brain organoids, with different simulation techniques, not only the one that I just described, but also high-density MEA that allows us to spatially target different areas of the organoids, or trying to play with the excitation inhibition balance to see if the learning mechanisms are improved. Another way, so this is extremely challenging because it's just trial and errors, but another way could be to use already designed, like we could use neuromorphic computing to train our organoid. And so this is a very nice collaboration that we have with the Professor Kalvenberg and the Professor Kuzum. The idea is to have on the same substrates, on the same chip, the recording of the organoids, and then this VLSI hardware, like the neuromorphic computation that allows us to do both signal processing and send stimulation, like a feedback, a reward feedback loop, thanks to the modeling of plasticity. Of course, I'm not an expert on this. If you have more questions, we can ask Gert. But I hope that I convince you that we have, even if we just made a few steps towards this new field, I hope that you see that we have all the pieces to do it, like to accomplish this goal. And I would like to thank my lab and the audience and all the collaborators.